Hello everyone, and welcome to Monica's Worm Corner. On today's episode, we're going to be talking about transcription and gene regulation. So transcription is the process in which a gene's DNA is transcribed into an RNA molecule. And gene expression is when a gene is turned on. And when I say that, I mean that a gene makes a protein that it specifies. So not all genes are turned on at the same time or in the same cell. And so what determines what gene gets turned on, when and where it gets turned on? Well, that's where transcription factors come in. So in eukaryotes, like this image that I've drawn behind me, eukaryotes need RNA polymerase and they need general transcription factors to bind to a target sequence in the promoter in order for transcription to be activated. So not all transcription factors are these general transcription factors. Some of them are specific to an individual gene. So that's just important to note for general purposes. But transcription factors have two roles, and that's to activate or repress transcription. And so if it's going to activate transcription, then that means that those transcription factors will allow RNA polymerase and or the general transcription factors to bind to the target sequence. If they have a repressor role, then that means that they will not allow RNA polymerase and or general transcription factors to bind. And because there is no binding, then transcription will not begin. And so humans are complex structures because we have trillions of cells. So how is it determined whether a gene is going to be turned on in a specific tissue? And so that's where enhancers and silencers come in. So an enhancer will activate transcription of a gene in a particular tissue or body structure. Silencers, like their name implies, will silence, so they will not allow a gene to be activated in a particular tissue or body structure. So transcription factors, some of them can be controlled, or some genes can be controlled by multiple transcription factors. And the way that this works can be in like a particular pattern type format. And so that's called combinatorial regulation. So I like to think, as an easy overall example, I like to think of a lock. So a lock usually has a 3-digit numerical code, and those numbers are arranged in a particular pattern. If you get one of the numbers wrong, or if you get the arrangement wrong, the lock is not going to open. So to think of that from a biological standpoint, let's say you have activator A, activator B, and repressor C. So activator A will only activate in skin cells. Activator B will only activate when there is divide now signals, and repressor C will come and kick in when the DNA is damaged. So in the presence of healthy DNA, the skin cells will divide. However, if the DNA becomes damaged, then the skin cells will stop dividing. And so the reason why this is important is because if the cell keeps dividing in the presence of damaged DNA, then cancer could potentially occur. And so what this shows is how gene regulation is this kind of complex structure that depends on multiple transcription factors that work together in like a pattern type format. And so this format is adjustable to the certain circumstances, like if DNA is damaged or if it's not damaged, because they're trying to mediate and decrease the negative consequences. So that was a short introduction into transcription factors, gene regulation, and gene expression. I hope you enjoyed, and thanks for watching.